From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on February 20th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 80th episode of Matters Microbial. Thanks as always for listening, viewing, and sharing the good microbial world. Should I make something special for listeners and viewers for our 100th episode? Please let me know. I am very aware of how worried so many scientists and science enthusiasts are these days. So here's a petri dish artwork created by my spouse, Dr. Jennifer J. Quinn, using fluorescent E. coli mirroring The Scream by Edvard Munch. And let me counterbalance that with some Lux art of my motto, Ovite Pauale Domini, which is Latin for All Hail the Small Masters. On a happier note, my Doc Martian research students went out and outfitted this plush tardigrade for my office, as you can see. They are very kind to me, and I appreciate it so very much. I recently put up a "what is your favorite microbe" question next to my lab down the hall, and sometimes I get odd answers from hallway passersby. But I sort of agree that Salmonella typhi and Mycobacterium tuberculosis are interesting choices. But favorite? In the next few weeks, I want to show listeners and viewers a little bit of what I can do with undergraduate students. Starting next week, I'll be looking for antimicrobial producing bacteria from lichens. After all, how do these symbiotic associations maintain the proper microbiome? Maybe through antibiotics. Here is a lovely example of lichens on a tree branch. In the coming weeks, I'll learn if isolates from lichens like these can inhibit bacterial growth as seen here. Wonderful zones of inhibition. And this is, of course, all based on the Tiny Earth program that we're so used to from Professor Joe Handelsman and co-workers. In any event, my life has been about teaching students to think like microbiologists. And I'd like to bring that to you. The amazing improvements in DNA sequencing and DNA analysis have revealed so many interesting aspects of the microbial world. From metabolites of interest, including antibiotics, to learning how different types of microbes coexist. It's a wonderful melding of standard microbiology and computer science, and it's the future. So I am delighted to introduce Dr. Marc Chevrette of the University of Florida to the podcast to discuss the work his newly formed research group is doing in this area and how microbes are the very best chemists. Welcome to the Quality Quorum for today, Marc. It's great to see you again.
Hey, Mark. Thanks for having me.
I'm delighted to see you. What's the weather like?
It's actually a little cold here in Florida. People are wearing jackets, and it's unseasonal, but probably nothing compared to what you have.
We have our 3 or 4 days of snow, and that's it, so I can't really complain too much. But it's been wet, I'll tell you that much. Which brings us back to lichens, I suppose. We met when I was visiting Joe Handelsman's lab, one of several times, and I was always impressed by the work you do, so I'm delighted you've decided to take the time to join us today.
Sure. Yeah, I'm really excited to share with you.
Yeah, and I was really taken by the first thing that you wrote to me, which is, microbes make the best chemists. And it's not something that people really appreciate initially. I often talk about this with students. If you look at an autotroph that gets all of its carbon from carbon dioxide, they have to build every carbon backbone for every biological molecule out of that CO2. And that's a lot of chemical tinkering.
It's a lot of chemical tinkering, but they've had about 1 billion to 2 billion years head start on us. And so they're pretty good at what they do.
Yes, they are. And it's an amazing thing. So the first thing that I'd like to ask is a little bit about what you mean by mining a genome, because some folks are novice micronauts here and some are more experienced.
Sure. So, genome mining, in the context that my lab likes to think about it, is trying to find out how bacteria are performing that chemistry through the information encoded in their genes. And so there's lots of complex chemistry across the tree of life, but the instructions for performing that chemistry are hidden in the genomes. And so now that we have all this deluge of genomic information, my lab and others have tried to figure out ways to look at that information and extract hypotheses that say maybe there's some new chemistry here. This is really important for the discovery of new molecules that could be antibiotics, for example, like you talked about in your intro.
What I tell people, and it's very interesting, and I'll talk a little bit about tiny earth, and you might have an isolate that gives rise to a zone of inhibition in several types of bacteria, and you don't know the mechanism of that antibiosis at all.
Mhm.
And most of the students I talk to think about how they would use chemical analysis to figure out what it is, like how big the molecule involved is by column chromatography, that kind of thing. But the other approach is to look at what genes are involved in to make the enzymes necessary to create that compound. Yes?
Correct. And oftentimes the chemical approaches are very informative, but they're also very slow. And some of them can be quite expensive. And so genomics enters the scene being an alternative that can get you to those hypotheses a lot faster and in some ways help you prioritize what's new and what's a boring antibiotic that may have been discovered before.
Right. And something else that folks, I don't, because we don't talk in undergraduate level stuff about the discovery of antibiotics, how they work, that kind of thing. That's why the cure is so important involving tiny earth. It's funny to me that it doesn't occur to folks that some of the antibiotics that we discuss might in fact be toxic to eukaryotes like ourselves. And
Yeah,
and
so that's very true, and that's especially true within the antifungals that we use in clinical applications. Oftentimes, when we look to nature, evolution has found a solution to some of these off-target toxic effects, and so we can see that in a couple of different systems. Many of them have to do with close-knit associations between bacteria and their host, where they're trying to fend off different types of pathogens, but also do so in a way that doesn't negatively affect their host. And so one of the approaches that my lab and others use to try and leverage that is to look within these systems. So how does nature actually figure these problems out? And can we use those principles in our own lab to as a springboard for discovery?
right. Now, one of the things that I talk about in classes is, and I know this will cause you a headache, just as we have the discussion of in vivo and in vitro, there are some folks who think that doing DNA analysis on a computer is in silico.
Hmm.
And the reason that I have a little trouble with that is I know many groups, including your own, actually do experimentation based on what they found.
That's correct.
And if you're studying the human genome, sometimes it's hard to do those kinds of experiments. And so I understand that. But what is especially attractive about what you and people like you doing this kind of research is that you can use these powerful tools to create hypotheses about how things are made and what they do, and then check it out.
Yeah, and I think that phrasing is really important. A lot of what we do as a lab on the keyboard is generation of hypotheses, right? That's not where the science ends. That's where the science begins. And so we take these large amounts of big data, oftentimes DNA sequences, trying to figure out these complex pathways. But at the end of all of that analysis, we're left with a hypothesis that needs testing. And so we go back into the lab and try and figure out what these molecules are, how they're working, and if they're a function of different interactions between species.
Parenthetically, and I think this is a good place to say it early on, you and I were speaking before we started recording about a dichotomy between people who do bioinformatics without bench work and bench people who don't do bioinformatics.
Mhm.
And you have some thoughts on how important it is to bridge that gap.
Absolutely. And people that come from both camps are making different assumptions in their analyses and in their experiments. And so being able to bridge that gap as a lab, I think, has situated us really well in that we understand kind of the assumptions that are being made in different types of analyses such that we can better design our experiments and vice versa. We can design our experiments to address some shortcomings or some lack of information that may exist from the analysis side. And so I think the field as a whole is embracing this, this kind of interdisciplinary crossing of different techniques. And labs like mine are filled with diverse people with diverse interests to try and straddle all of those different areas towards the goal of finding this new chemistry.
I've said it many times. Different faces and voices and brains are really important in science. And there's a saying I heard, and it was assigned to a culture. It's not important which one it was, but that the nail that stands up gets hammered down, and that creates a uniformity of view. And I still maintain, especially when you're out on looking at things that haven't been well studied, that those kind of outside the box ideas can be very important. And you can get fooled very easily. I remember, and I don't know if I've shared this story with you many, many years ago, and I mean no disrespect to the people who did this work, but they were saying, for example, that the predatory bacterium Vibrio did not have an ATP translocase as hypothesized. That is to say, it wasn't taking ATP from its host into itself to power its metabolism. And they said that because there were no genes in the genome related to the ATP translocases that they knew. Problem was, people had studied ATP translocases in Vibrio and published on it. And that's a really good example of bringing together that wet lab and bioinformatics part.
Absolutely. And that draws some attention to one of the problems we often find in our analyses is that you're often really good at knowing how to look for things you know how to look for, right? And
Yeah.
so if discovery is your goal, we kind of we've adopted the mantra of fail often, fail fast. So be loose with our hypotheses on the keyboard so that we can be really strict with our validation on the bench.
Fail often, fail fast. It sounds terrible, but that's how you progress.
Absolutely.
It's like I say in the classroom, I don't know isn't a sign of ignorance, it's an opportunity, right? To learn new things, to have different ideas. So it's really interesting you bring that up, because this is what we need to be able to do. So as I understand it, because this is the stuff that at my level anyway, often if we're looking at a genome, when we have, sometimes it's wholly automated these days, annotation determining what certain open reading frames are encoding, possibly based on their relatedness to known genes. And now you're looking beyond that now at like networks of genes.
That's correct. Yeah, oftentimes the types of chemistry that goes into the creation of these antibiotics and other types of molecules we might steal and use medicinally are quite complicated. And so these are typically many genes, sometimes 2 or 3, 4 dozen genes that are all working in concert to coordinate the assembly of these molecules in a very precise manner. And so because these instructions are encoded within the genome, and we can see that many of these bacteria have different phenotypes, like say killing another bacterium, producing a zone of inhibition, we can start to connect those dots if we leverage data from our experiments and try and marry that to the genomic information. Again, trying to kind of pinpoint those mechanisms. So why is this phenotype actually happening? What are the genes involved? This is very much speculative and a hypothesis, and so that requires further validation on the back end. But we have so much data now as a field. People are genome sequencing faster than they can get their cup of coffee in the morning. That presents a different challenge. The challenge now isn't getting the information, it's making sense of that information because there's so much of it.
You know, the joke is drinking from the fire hose, right? Like that whole
Exactly.
meme. And I remember, because I am ancient beyond words now, when getting 300 bases on an acrylamide gel DNA sequencing was like a big deal.
Oh yeah, that could be an entire PhD just a couple of decades ago.
Yes. And so
Mhm.
now this is the stuff that you send out and you can get, well, a lot more than 300 bases the same day in some cases.
Yep.
And so that's transformed the field, as you say. One thing that always surprised me, and maybe the right way to go about this, is to use a particular example of an antibiotic if you care to. I look at a lot of antibiotics having quite complicated structures.
Mhm.
And these are examples of secondary metabolites, things that microbes often make, usually late in their life cycle, for lack of a better term, and they can have these biological effects. I mean, you did your PhD work on similar sorts of things and involving fungus farming ants.
Correct.
And so what would you do to identify, say it? Let's just say that in this example of the zones of inhibition that I showed you against a couple of different bacterial taxa, what would be your next move to figure out what that antibiotic might be?
Well, one of the first questions I'd ask is what is the bacterium that's actually getting killed? And what do we know about it? So are there any mechanisms of resistance that have been studied in that bacterium? And can we use that to kind of guide our search within the genome? Oftentimes, the bacteria that are producing these molecules are not producing one. They're producing many dozens of molecules. And so even though we can go into the genome and find these pathways, we're still left with a challenge of figuring out what is the causative mechanism for that interaction. And so trying to leverage all of the data that we have, and most of that data is getting generated through bench experiments. So how are they getting killed? Are there different morphological changes that are experienced? Is there some sort of pigmentation that is diffusing across the agar? All these different things that you can see visually, or perhaps more data centric through transcriptomics or metabolomics. Are there signatures in data that we see in these interactions that are absent from when these bacteria are grown alone? And so that's a lot, but integrating all of those different things together kind of helps us prioritize not just the bacteria to study, but which pathways within those bacteria are likely to give us the result of a new discovery.
And evolution always uses the same general sets of tools and equipment to do things. And so often, if you understand one pathway to make a particular secondary metabolite, if there's another secondary metabolite with a similar structure, perhaps those genes are involved in its synthesis as well.
That's largely true. I think that's been the basis for many of the genome mining approaches that have existed in the past. But I will caveat that with evolution surprises us every day. And so there are definitely new discoveries to be made on the evolutionary side of things. And that's where a lot of my lab has been focused on in the last couple of years. So trying to leverage evolution in ways that may not fit the paradigms that are well studied, but in the emergence of new genes with new function and how that plays into making new chemistry.
As you know, I have a colleague in my department here who studies bacteria that make antifungal agents. And in doing very, very simple bioinformatic analysis, what you said before really struck me. That's very clearly more than one molecule involved in this.
Yeah,
And
and oftentimes they can work in concert with one another. There are some types of molecules that can affect the cell boundaries of the target organism and make it easier for an antibiotic or an antifungal to cross the membrane. And so that's a case where an antibiotic or an antifungal may not be the only player in the story. It may need
right.
some other supplement to synergize with and then see the activity that we see either in nature or in the lab experiments.
It's
So
so
it is
in,
quite complicated.
it's so interesting to me how reductionist we are. You know, one gene, one phenotype, and that was hammered into my head so many years ago. And it's simply not true. It's true for particular model systems under particular conditions. I don't know if you heard the joke that Esther Angart said to me a couple of podcasts ago that I like so much. We were talking about the Minot comment that what's true for elephants is true for E. coli. And her response was, what's true for E. coli is true for E. coli some of the time.
Yeah,
And
and E. coli is E. coli is an interesting bacterium to bring up because as a species, there's so much diversity within that just one very small, narrow lineage of the tree of life that even within E. coli, we see half, sometimes more of the genome that's different between different strains of E. coli. And so what's true of E. coli may not even be true of E. coli, depending on what strain you're using.
it's just amazing to me and how we give those names. My point is something we've talked about on the podcast many times are the interrelations between organisms
Mhm.
and how to tease those out. And we have so many new tools that are available, and genome mining is one of them. So you've looked at organisms that make one particular type of antimicrobial compounds, and you've analyzed those by doing both the computer work and the wet work. What about some more complicated things that your lab has looked at?
Well, I think one of the questions that we're often left with at the end of that is why? So why are these molecules getting made in the first place? And perhaps an even tougher question is how we can elicit them to be produced under laboratory conditions. Because if we're leveraging evolution to guide our genome mining, these molecules and the genes that encode them are are functions of that evolution. And when we bring them into the laboratory, we're kind of divorcing those two ideas. It's a very artificial environment, one that oftentimes these bacteria are not going to interface with in nature. And in these natural systems, they're almost never alone. They're part of very complex communities, sometimes spanning 5 to 6 orders of magnitude, interacting with each other at both local scales and at a distance. And if you introduce a host, like in the case of a microbiome, that complicates things even further. And so what my lab has tried to do is kind of take this real big, sticky, hairy problem and build from the bottom up and try and understand that through making synthetic communities where we can see those interactions happening, both in terms of like actually seeing them in the case of pigmented products, but also see them in terms of things that are more difficult to see visually. So I'm talking transcriptomics, which genes are going up and down, and metabolomics, which metabolites are produced under certain conditions or in certain combinations of bacteria that you wouldn't see when they're growing happy in media all alone in the lab.
I think that's exactly right. And again, for listeners and viewers, transcriptomics is where you study the messenger RNAs that are being made under different conditions. And sometimes you can come up doing this with whole genomes, something I could never have imagined. I've done it in my own little lab where you'll be able to say, okay, under this condition, here are 15 genes that are upregulated by 2 to 3-fold. And here's another 20 that are downregulated by 2 to 3-fold. And what you do with that information, as you've said throughout our talk today, becomes important. Metabolomics the same way by studying small metabolites that change their concentrations at different times, different conditions. And again, it's a beautiful time to be a microbial scientist for that reason. We have these kinds of tools.
And in the case of the biosynthetic gene clusters that I'm interested in, that's especially true. There have been some estimates out there that say that almost 80% of these gene clusters that can make these types of molecules are silent under laboratory conditions. And so we know that they're important because they're conserved over evolutionary timescales, but we don't really know why they're important. And that kind of question of why is one that really drives some of the interaction work in our lab. We think that a lot of the explanation for that is the community context in which these things have evolved and in which they thrive and flourish. And so if these molecules are evolutionary products that are mediating interspecies interactions, what we're trying to do as a lab is to try and better understand those interspecies interactions, to see if we can get them to make things that they wouldn't otherwise.
Did you ever have a chance to meet Julian Davies?
I did, yes, and actually, Julian's work is inspiring us every day. He's definitely one of the early people that kind of instilled this idea of antibiotics being a very human word, a very human context. And antibiotics in nature may not actually function as antibiotics. They could be signaling molecules. They could be mediating all different types of interactions depending on their concentrations.
I'll put some links in the show notes to this, but Julian Davies was a wonderful person. He didn't care who you were, at what level you did research or taught. He would sit and spend time with you and talk to you,
Mhm.
and he was extremely enthusiastic, and I had the best time. Number one, I learned in the development of antibiotics, it's almost like, I use this term very carefully, almost like black magic, figuring out what to add to the culture conditions to maximize antibiotic production. And he told all kinds of great stories about it. But the second issue is the one you allude to, the possibility that antibiotics could have a very different effect in nature than we think they do, because we use them therapeutically at much higher concentrations than they occur in nature. And that was transformative for me to think about.
Absolutely, and some of his early work showed that that's exactly the case when you look at sublethal concentrations of antibiotics and the types of regulatory craziness they can cause in other bacteria that experience that sublethal dose of antibiotics.
You
Um,
know, I was just going to say, I was sitting having lunch with him one day, and he was bemoaning the fact that there were so many omics out there, and he didn't have any omics to study. And then because he was interested in small molecules, I told him, because I had come up with this bad Latin motto from, for what my interests are. And I said, well, parvus is Latin for small. So how about the parvome? And he published several times calling it the parvome, that constellation of small molecules that we tend to ignore.
mhm.
So you're interested in looking at, and again, I was very taken by what you were saying from the study of biofilms, where people would study biofilms using one organism.
Mhm.
And then I would say, well, that hardly reflects what one sees in nature. And they said, okay, I'll use three. And I understand where that comes from. But the fact of the matter, in nature, things are so much more complicated. Sometimes you have to start with the one or two or three things together to get to the interesting questions, but it does not reflect nature very well at all. And that's what fascinates me. And I haven't even thought about the temporal nature of things. So there you
Yeah,
go.
often these interactions are quite dynamic, right? And they're dependent on different things. So that themselves are dynamic. So the different ratios of bacteria, the opportunity for them to physically interact, different changes in things like temperature or humidity or different concentrations of nutrients. These are things that are often in flux in nature. They're not static, but in the lab we have them fixed. And so it's our contention and one of many other labs that are kind of pushing in this direction that we should go back to nature. We should try and figure out what's actually happening within these dynamic systems, and then use that to inform how we design our experiments.
I think so. And even with first year students doing a little bit of a cure, like finding out, please forgive me, what microbes grow in their water bottles.
Mhm.
We've found microbes in those water bottles that are making antimicrobials. And maybe they're doing that to free up real estate in that water bottle or signal. I mean, we don't know. It's a very artificial situation.
Yeah, and as we get more and more genome sequence, it's becoming clear that the capacity to make an antimicrobial or these different types of chemistry is not unique. It's something that's shared across the tree of life. Now that chemistry can be very, very diverse depending on which microbe we're talking about. But the ability to make it is not something that makes them stand out. It seems to be the rule across nature is that these molecules are quite common. And so yeah, that leaves the question, why?
I mean, that's the thing that excites me every day to think about, what I'm going to see in the next week. Mark, when you're looking for organisms that produce antimicrobials, there's an issue of antibiotic resistance being a growing problem in medicine with organisms that cause disease. Do you see evidence, even in this process, of certainly other bacteria becoming resistant, even at that early stage? And also, are there any thoughts about how the organism that produces the antimicrobial is resistant to it?
Sure, sir. I'll tackle that in backwards order. So the microbes that are producing antimicrobials have to figure out a way not to kill themselves. And so they're by definition need to be resistant to these types of molecules. And that's guided a lot of our genome mining efforts. There are some of these pathways that have resistant elements embedded right within them because they need to be co-transcribed in order to make sure that they don't kill themselves. But what we're finding as we're looking across the tree of life, especially within different bacterial lineages, is that even though that's a very logical way for a microbe to make itself resistant against what it's making, it doesn't seem to be the rule. Oftentimes, these pathways do not harbor resistance. And so that leaves an open question for us in that how are they avoiding killing themselves? And so we're taking a few different artificial intelligence and machine learning approaches to that to try and leverage whole genome information. So things that may not necessarily be clustered with the pathway of interest, but maybe off that locus and acting in trans. So acting at a distance. This is a very simple question to pose, but a very difficult one to answer. And so we're trying to leverage big data to be able to do that. In terms of resistance in the organism that you're trying to screen against, the one that you're trying to kill that might be a pathogen of interest, for example, we've taken a few different approaches. The different libraries of bacteria that we're using in these tests themselves are picked to have different types of resistant mechanisms embedded within their genomes. And not only that, they're genetically tractable. So we can go in and assassinate the capacity for that resistance to see if we can, if we can find ways to either exacerbate or relieve the antibiotic burden that we see within these pairwise studies. And so very much kind of like a reverse genetics classical approach to taking genes away and seeing how that affects things to try and get more towards mechanism. And what we're really interested in are the ones that, when we make those perturbations, still work, suggesting that there's a new mechanism of resistance or a new mechanism of action that hasn't been described before.
And that's the greatest feeling, is to find something that no one has seen before.
Absolutely.
So I think, we've talked about some simple things. Are there specific projects in your laboratory that you'd like to talk about?
Sure. So, kind of to springboard off of that introduction on microbial interactions, our lab is trying to figure out a way to leverage chemical interactions between species in a way that we can build a high throughput screening pipeline to see what types of chemistry they make. Forgive me for taking a reductionist approach on this, but we've put together a synthetic community of about 12 to 15 microbes, depending on how you count, that all interact with each other in certain ways. With that, we've been able to figure out how they change each other's morphology, how they change each other's developmental cycles, so things like sporulation or the formation of aerial hyphae. And now where we're going with that is trying to better understand how chemistry plays a role in mediating those interactions. Because we know from prior work that microbes in communities behave very differently than microbes alone in terms of their chemistry. And so we're trying to use that principle as a way to generate new chemical diversity that we can use to maybe look for new antibiotics. So if bacterium 1 and bacterium 2 are together on a plate, maybe that there's some interaction that triggers the production of a molecule that might kill bacterium 3. Or maybe it's a new antifungal that eludes the toxicity issues that you talked about before. And so we think this is closer to nature than the way that this has traditionally been done in the past. The whole kind of like black magic of elicitation, we're trying to put a finer point on that and seeing when and why molecules are actually produced. And a lot of our interest is around how bacteria are talking to each other in those conversations, how that plays a role in that elicitation of chemistry.
It's so funny when students get very little, and when I say this, I want to be very cautious. I mean, no critique of anyone when I say this, but when you're at a smaller institution anyway, and I presume this also happens at larger ones, you don't get a lot of microbial science in the standard biology curriculum. And when the students learn, for example, about quorum sensing, they have a very limited view of that chemical communication modality. And what I try to tell them is, microbes are talking all the time. And I think there's a Letterberg quote about that we just don't know the frequency. And I think that might be true. But the fact of the matter is there's huge conversations going on a microbial level everywhere you look. We're just not equipped to hear it. But the way that they do it is not acoustically. They're using chemicals, as you mentioned. And sometimes, and this is something that's been ascribed either to Leslie Orgel or to Francis Crick, that evolution is smarter than you are. And it's really true that often we make assumptions about things based on what went before instead of first principles. And
evolution often takes that fail often approach.
Yes. And in fact, you can show evolution occurring in the laboratory by passaging bacteria in a week. Mm-hmm. Right? So you can easily show that the hand of Darwin is on everything that lives. It's absolutely true. And that colors, I have to be careful when I teach that because then students don't believe anything. And I don't mean to cause that to happen to them. But when we look at one organism in pure culture, that's not how they normally live. Any more than if you took one person and plunked them down somewhere by themselves. It's the same kind of principle.
And it's funny you bring up Darwin because I often in my introductory classes introduce microbial chemistry by using Darwinian finches. And so the morphology of their beaks really is an evolutionary product of how they interact with their environment and how they can be fit in their local environments. And the same is true of microbial chemistry. It's just a little harder to see.
I want to say, and for listeners and viewers, did you work on the Thor system? I'm not recalling. Did you?
Yes. Yep.
So as part of the Thor system, you have like 4 organisms and huge amounts of interactions and changes with just 4.
So we've got there's 3 in the traditional Thor system. There's a bacillus, a flavobacterium, and a pseudomonas. And one of the more interesting things that has come out of Joe Handelsman's lab in the last couple of years, and some work that myself and others helped on, shows that when they all interact in a 3-member community, a large part of the chemistry and the genes that go up and down are only seen in that 3-way interaction environment, rather than what you would see with the microbes alone or the microbes even in pairwise. And so if you take the information of the microbes alone or the microbes in pairwise and try and make an additive model to say, does that explain what you see in the 3-member community? It often fails. And the dominant explanation of that information is in that 3-way interaction term. And so that's, even in these like quite simple reductionist approaches, we see emergent properties that are happening in the simplest of communities.
That is exactly the term I have written down to bring up with you, that emergent properties are the things that you don't expect based on what went in. And it's, to me, one of the most interesting things about all this. And so when you have, say, a dozen organisms in one of your communities, there's dizzying possibilities even with that.
Right. And one of the things that we use as kind of a null hypothesis is trying to figure out what the one-on-one interactions are and seeing where they break down when you introduce this greater complexity.
Yeah, I think that's great. And I've seen tons and tons and tons of things over the years where, forgive me, a colony on a plate here, a colony on a plate there, and where they're closest, you'll get pigment in one of the organisms and not the other.
Mhm.
just as a very simplistic view of the kinds of communication that's taking place.
Yeah, and,
Um,
and we're trying to leverage that at large scale in our lab. So I've got a few pictures to show you that kind of put some
Please.
color into those colorful interactions. One of the bacteria that we're interested in, because it's been so well studied in terms of its metabolism and its genetics, is Streptomyces silicolor. It's called Streptomyces silicolor because it produces these beautiful blue pigments, but it doesn't do so all the time. And often what we're finding is that the production of these pigments is a function of whether or not they're interacting with another bacterium. And so
mm,
when you grow them with another bacterium, they may make more pigment, they may make less pigment. And remember that these pigments underneath are chemistry that itself has different function and can perhaps be antibiotics. And so it's a really nice visual representation of what's happening at the micro scale. But of course, remember that this is just one of many different molecules that are probably differentially expressed under these conditions. And so we see this as a proof of concept that's very visually striking, but we're also really interested in the stuff that you can't see visually. So the
mm.
molecules that are being produced that you can only see through new omics methods. So things like liquid chromatography, mass spectrometry, for example.
That's amazing stuff. And you're absolutely right. I've seen even with my own work with undergrads when they will do a survey of the kinds of organisms that they see in, say, soil or off lichens. There's a lot of pigmentation, and you have to ask yourself, what is that pigmentation for?
Mhm.
And some of the time, I think it's about communication. Some of the time, it's about protection from UV, and
Mhm.
some of the time, it's about trying to keep your real estate going.
Absolutely. And, you know, all of those different options are equally good hypotheses, but another is something we haven't even thought of before, right? So understanding new mechanisms and new function. Um, like I said, evolution really surprises us every day. So,
It's wonderful. So I really like this idea, and streptomyces is a big favorite of mine. I know a person, and I'll try and find a link to her for the show notes, who actually does artwork with the blue pigment from Streptomyces silicolor.
that's great.
Yeah. Mark, in your discussion of coming up with pairing microbes to study them, I wonder if you could tell listeners and viewers a little bit about how you do that. Is it like something where you're using a database, several different isolated organisms? I mean, how do you develop the idea of how to pair them together?
So we're taking two general approaches to address that question. One is in our purely synthetic community, we've filled that community with bacteria with at least one known interaction with another bacterium in that community. And so of those 12 to 15 that I mentioned, they're at least shown once to interact chemically with another member of that consortium. And so that's one way we're doing it. So kind of this bottom up building approach. But another is looking to nature and in natural communities that have been assessed either through 16S rDNA amplicon sequencing or through shotgun metagenomics, where we know the who of the different bacteria that are there. Using co-occurrence networks and different methods of machine learning to try and understand the patterns of co-occurrence of microbes that may suggest that they're going to be interacting because they commonly co-occur. And so we're kind of taking those two approaches. One, I think we tend to call bottom up, and so building from interactions that are known and trying to layer complexity onto that. And one that's more top down, so taking a really complex community of microbes, let's say on the skin of amphibians, and seeing which microbes co-occur with one another. And then from there, narrowing down our list of hundreds of thousands of species to a few that we think are going to likely interact.
I was thinking about that a little bit as you were saying it. There was this horrific report that if you drink a lot of coffee, there's a specific bacterial taxon that appears to kind of conquer your gut. So this is something I clearly have to look at since I drink way too much coffee. That's a great project, and I really appreciate the things that you've talked about with it. Are there other things in the lab that you wanted to share with us today?
Yeah, so one of the major questions we have in our lab is how does do the principles we see in our synthetic community actually translate into real world natural communities? And so we're taking kind of these approaches of pairing bacteria together, either one-on-one or in more complex communities, and trying to inform that by the frequencies we see them in more natural communities. And so a few of the systems that we're really interested in are the skin of reptiles and amphibians. And we really like those because they've been bombarded with fungal disease over the last 2 to 3 decades, in a way that, at least for amphibians, has caused over 100 different species extinctions at the amphibian level. So there's a mass extinction going on that a lot of people aren't aware of that is killing frogs and salamanders that is a fungal-borne disease. And there's been a lot of research to try and understand how the immune system of frogs and salamanders contributes to mitigating that disease. But we think as a lab that the microbiome plays a key role. And so the skin and the mucosal microbiome of these different amphibians and snakes harbors bacteria that themselves can make antifungals. And like we talked about before, they have to do so in a way that doesn't negatively impact the host, or at least in a way that the host can tolerate. And so the mechanisms of that are still elusive. We're still in the very early days of trying to figure out how, when, and why this is happening. But we're taking similar approaches. And so looking into frog microbiomes, seeing who's there, and trying to isolate them out, and then building mock communities to see how the chemical interactions between them may be contributing to antifungal production.
You know, it's an interesting story with the chytrids and Genethobacterium,
Mhm.
which I was raised to call Chromobacterium, but you know how it goes. But what's interesting there is that the way it's portrayed in kind of the science communication is very simplistic. And I'm
Mhm.
delighted. It sounded judgy. I don't mean it to sound judgy. I think it's a great thing because I like amphibians a lot. I couldn't have dogs or cats growing up, so I had a lot of critters.
Mhm.
My point is thinking about how an organism can defend itself against a potential disease-causing organism is important. And I doubt it's just one organism, but it's groups of organisms working in concert that can do things, which shouldn't surprise me, right?
That's correct. And oftentimes you need to have multiple shots on goal to be able to mitigate these diseases because remember, the pathogens themselves are evolving, right? And so
Yes.
you're in this kind of race of fitness. Is the pathogen going to win, or is the host and its associated microbiome going to win? And that kind of back and forth match often plays out over chemistry.
I hadn't heard about the snake business. This will be very interesting for me to follow up on.
So it's a different fungal disease, but it impacts the snake in the same ways as the chytrid disease in amphibians. The snake skin environment is a really interesting one because it's extremely keratin rich, and keratin isn't really found in too many other places. So we see a very unique microbiome on the skin of snakes that you wouldn't find in surrounding soil, for example, because the local environment is so strange. It's got very different lipids that you would see in other environments. And we've shown through collaborations with Donnie Walker at Middle Tennessee State, who's really led this research, that the chemistry of the lipids on the snake's skin can predict the microbial community, and the microbial community can predict the lipids on the skin. And so that kind of interplay for us is really exciting because all of that together seems to play a role in whether or not the snakes are going to be susceptible or resistant to the disease. And so we're looking in now in our collaboration with the Walker Lab to try and better understand that. And so what is actually happening in terms of the chemistry? Is that chemistry producing antifungals only in the community, or is it something that is a more simplistic system like the Xanthinobacterium, where you have one bacterium, one molecule? Um, we're still ongoing work. It's still ongoing work to figure that out. But it seems like it's a more complex story within this system, and it's a really exciting one to study.
Well, I have a personal interest since I walk around the hallways with my pet Kenyan sand boa quite often. I
Hmm.
will never look at, and I have had students ask me, could they swab that poor little boa? So now we find out there are people
Sure.
who do exactly that kind of thing.
Well, if you want to get it sequenced, you know where to send it.
That would be kind of fun to think about, wouldn't it?
Mhm.
I think this is wonderful. And as we're starting to wind down in time, I think it would be good for you to talk a little bit about a topic that always kind of takes me back or makes me a little nervous. It's the concept of big data. And how did you come to your melding of wet lab with what I'm going to call big data simply because of the amount of genomic sequence you have to look at? Was that an evolution, or did you start in one area and end in the other?
Well, I started my undergrad training in a program that was brand new. They called it bioinformatics and molecular biology, but really it was those terms weren't well defined at the time, and so it was half computer science, classical computer science training, and half molecular work. And it always kind of frustrated me that those were in two very discrete buckets. And so the push for me in my subsequent training has been to kind of bring them together. And so not have them as kind of these distinct disciplines, but as a straddle of the two, such that you can ask big and interesting questions. And that's what really led me into microbial chemistry as one of the more interesting questions, because as you sequence and you get more and more data, oftentimes the technical problems that you get with genome assembly have to do with things like high GC content or repetitive regions. And when you see these things over and over again, you start to ask, well, what are these regions? Like, why would nature make my life so horrible as a sequencer? What's going on here? And that led me into microbial chemistry, because the repetitiveness and the high GC nature of a lot of these different biosynthetic pathways makes them difficult to sequence via traditional methods. And so that's largely a solved problem now with the onset of long read sequencing, where we can span really large regions of DNA in one go. Um, but the interest and excitement around these pathways remains. So even though we can see them relatively routinely now, we still have to figure out what they do and why they're there.
Yeah, and as I understand it, and we've talked about this in other venues, there are certain programs that help you look for possible biosynthetic gene clusters, which
That's
is the
correct.
which is the center of this repetitiveness that you're mentioning.
Hmm. Yeah, and so a lot of those tools have been widely used for the last 10 years, but they're often built for a very particular question. And so one of those questions is, if I have a genome, what's in it? And so that's a very valid question and one that people ask all the time. But what my lab has tried to do is develop some other tools to ask different types of questions. And so if you flip that and you say, oh, I have a pathway of interest, which genomes is it in? That's a very easy question to ask, but a more difficult one to answer computationally. And so we've built some pipelines to try and address that.
So it's interesting to me, and we talked a lot about bringing these things together. I've sent a fair number of students off to graduate school and
Mhm.
earning their PhDs, and some of them have become bioinformaticians. What advice would you give people that are going to graduate school, or perhaps even what should be offered in the undergraduate curriculum that would help prepare people for this ability to kind of meld both the computer science and the wet lab business?
Well, I think a large part of it has to do with really fostering your own curiosity. I think one of the things that bioinformatics and computational work does quite well is it enables you as a researcher to be able to ask many more hypotheses, many more questions than you would be able to do with the limited amount of time you have on the bench. And so, you know, being able to really ask those kind of questions that are driven from intuition you get on the bench and on the keyboard, I think is where people really succeed in this area. That's
Yeah.
something that I try and instill in my trainees, and they've done an amazing job to be able to, take these ideas that they have and then have the tools to be able to put some either validation or form hypotheses around these ideas. And so I said at the very beginning that these conversations kind of between the pure computational people and the very classical microbiologists can sometimes have friction because they're coming from different places. And so being able to speak the language of both is something that I would encourage new people into the field to be able to do, because it really allows you to be able to be a truly independent scientist, but also one that forms lots of collaborations with people from that span different disciplines.
Something that you're saying reminds me of something that when students take statistics, they'll often tell me when they're advisees of mine that they don't like it, which
Mm-hmm.
always amazes me because they're much better at mathematics than I ever was. But what's interesting to me is that when they take it in the context of answering biological questions, because we have such a
Mm-hmm.
course here, those same students love that.
Absolutely.
And that's exactly what you just said, is that you have to have that buy-in. And, some people just love the numbers and the number crunching. Other people need to have that kind of connection to another question.
Mm-hmm. Yeah, and I think that's what sets bioinformatics apart from a lot of different types of computer science, in that it's really driven by those biological questions. Like you can see the carrot at the end of the stick, right? Like you're not crunching numbers for the sake of it. You're crunching numbers towards a goal, which is better understanding and insight into the biology. And so yeah, oftentimes statistics and more basic math is taught in the abstract. But I think what we're kind of developing as a field is now that bioinformatics is more of a standalone science, that trainees are getting to see how those applied questions are actually coming into where you use statistics or you use code, not in the abstract, but in the actual application of answering your question.
That's been very, very useful. Mark, I've really appreciated this opportunity to talk with you about your research program, the way that you've created a modality of bringing different ideas together, the really quite complex questions that you're starting to dive into. It's been very interesting to me, and I hope to our listeners and viewers.
Yeah, and thanks so much for the invitation to share my work with you. I think this platform is an awesome one, and I hope that I've inspired some people to start looking at microbial chemistry.
And because who are the best chemists?
It's not me.
And I would say very probably the oldest chemists.
Very much so.
My very best wishes to you and your lab group.
Thank you, Mark.
You're welcome. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episodes, with wonderful links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Marc Chevrette can be found in the Department of Microbiology and Cell Science at the University of Florida in Gainesville, Florida. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.